Okay, so in today's lecture, we will look at a very important disease called cancer, and this is under the umbrella of anti-cancer agents. So, before we go into the anti-cancer agents, let us try and understand the disease itself. So, cancer is one of the most devastating diseases worldwide. According to the World Health Organization, 1 in 3 are actually affected by this. And it is also found that about a quarter, that is 25% of all deaths in the developed world during the year 2000 is due to cancer. In India as well, cancer is becoming a huge problem, and the number of deaths due to cancer is actually going up. Of course, infectious diseases still take a large toll on people, but as time has progressed, cancer has also become an important disease to tackle. And worldwide, it is only second to heart disease in terms of the cause of death. So, cancer cells are formed when normal cells lose the normal regulatory mechanisms that control growth and multiplication. So, these cells become what are known as rogue cells. So, rogue cells are nothing but cells which are not going to follow the rules. Okay. So, this process is called as loss of differentiation. So, typically when a cell divides, and then what happens is that it goes through a process called as differentiation. So, differentiation means from the stem cells, what happens is that let us say all our cells are derived from germ cells or stem cells. And so, at some point these cells differentiate and become bone cells or muscle cells and so on. So, in cancer, this process is accompanied by a loss of differentiation. So, the term neoplasm is actually used to indicate new growth, and is actually a more accurate terminology for the disease. The more common names that are used are cancer and tumor, and tumor in reality actually just means a local swelling. So, if the cancer is localized, it is said to be benign, and if it invades other parts of the body and sets up what are known as secondary cancers or secondary tumors, this process is called as metastasis. And such cancers are defined as malignant. So, the benign cancers or benign tumors are actually not a major problem. The malignant ones are the ones that become life threatening. So, a major problem in treating cancer is that it is not a single disease. There are over 200 different types of cancers resulting from various cellular defects. And so, a treatment that is effective in controlling one type of cancer is completely ineffective on the other. So, therefore, we need to evolve multiple strategies to address this disease. So, as many as 30% of cancers are actually caused by smoking, while another 30% are pretty much diet related. So, there are a number of molecules which are known as carcinogenic molecules. So, what this means is that these are cancer causing chemicals, and these can be in the smoke or the food and of course, the environment. And what these can do is they can induce gene mutations or interfere with normal cell differentiation. So, once one of these two mechanisms happen, then it is likely that cancer can develop. So, the process of the birth of a cancer is called as carcinogenesis, and it is initiated by a chemical known as a mutagen. But other triggering events such as exposure to further mutagens, for example, smoke from a cigarette for example, are also required for the cancer to develop. There are some viruses which have been implicated in at least 6 human cancers, and these cause about 15% of the world's cancer deaths. Okay. So, for example, the Epstein-Barr virus is the cause of Burkitt's lymphoma and nasopharyngeal carcinoma. There is another kind of virus known as human papilloma virus, which is sexually transmitted and can lead to cervical cancer. There is another virus causing hepatitis B, which leads to 80% of all liver cancers. And of course, HIV can cause Kaposi sarcoma and lymphoma. So, one major way in which cancer can be initiated is through the presence of mutagens, and these mutagens can cause genetic defects which can be further accentuated by continued exposure to the particular mutagen. Or the other way in which cancers can be initiated is through viruses. Now, virus can actually bring about cancer in several ways. So, they may bring what are known as oncogenes. Oncogenes, we will look at it later, but these are genes that are important in cancer, right. And these oncogenes can be brought into the cell and they can insert themselves into the genome. Just a couple of lectures back, we looked at how the virus integrates its own nucleic acids. It could be RNA, which then further is converted to DNA, or DNA based viruses can actually go and insert themselves into the genome. So, for example, the Roux sarcoma virus carries a gene for an abnormal tyrosine kinase. So, some viruses carry one or more promoters or enhancers. Okay. If these are integrated into the cellular genome, then the promoter stimulates its transcription, leading to cancer. Cancer can also be sort of initiated by bacteria. So, the famous case is the Helicobacter pylori, which is responsible for many of the stomach ulcers and is implicated in stomach cancers. So, the treatments used to combat cancer, which are typically radiotherapy, where high intensity radiation is used, or chemotherapy, wherein drugs are given, can actually induce a different cancer in surviving patients. So, that is one of the problems in using these types of therapies. However, these methods can also be used to treat cancers. For example, 5% of patients cured of Hodgkin's disease developed acute leukemia. Nevertheless, the risk of a second cancer is outweighed by the benefit of defeating the original one. So, some patients are prone to certain cancers for genetic reasons. So, it is possible that certain damaged genes can be passed on from one generation to another. Okay, so, the most common example is in the case of breast cancers. And so, the presence of these genes can lead to an increased risk of development of cancers in subsequent generations. So, now we will look at the concepts of proto-oncogenes and oncogenes. We have already looked at been exposed to the term oncogene. So, proto-oncogenes are genes which normally code for proteins involved in the control of cell division and differentiation. Okay. So, these normally code for proteins which are involved in the control of cell division and differentiation. Just to remind you, cell division is the process by which cells divide, and differentiation is the process by which a stem cell gets converted to a cell with a function, for example, a muscle cell. Now, when these proto-oncogenes are mutated, this disrupts the normal function of the cell and the cell can potentially become cancerous. So, then this term proto-oncogene is defined as actually an oncogene. So, one example is the RAS class of oncogenes. So, this gene codes for a protein known as RAS, okay, which is involved in the signaling pathway leading to cell division. In normal cells, this protein has a self-regulating ability and can switch itself off. However, if the gene becomes mutated, an abnormal RAS protein is produced, which then loses this ability and is continuously active, leading to continuous cell division. So, when you have an abnormal RAS protein, then what happens is that you would have the cell dividing continuously, or it becomes immortal. It has been shown that this mutation of RAS gene is present in about 20 to 30% of human cancers. So, these oncogenes, as I discussed earlier, can also be introduced by viruses. Now, there is another concept which is known as anti-oncogenes. So, these are nothing but tumor suppression genes. So, here in a normal cell, if you think about how during normal metabolism, it is possible that DNA is damaged. For example, we go out in the sunlight and some of our DNA gets damaged. Okay. But there are policemen in the cell which can actually detect these damages and then block the DNA replication, so that you do not have faulty division happening. So, once you block the DNA replication, it gives the cell enough time to repair the damaged DNA before the next cell division. However, if this cell repair is not possible, then there is a series of events that is triggered in which the cell actually commits suicide, and this process is called as apoptosis. So, there are genes which are known as tumor suppression genes, and these actually code for proteins that are involved in the process of checking whether there is damage in the cell or not, repair in the case of DNA damage, and suicide. So, this is a very intricately regulated process, and tumor suppression genes are forming an important part of this. So, an example is this is TP53, which is a gene and which codes for the protein by the name P53. Okay. So, P53 protein is very intimately linked with several cancers. So, if the TP53 gene is damaged, then the repair mechanisms become less efficient. So, once the repair mechanisms inside the cell become less efficient, the defects are carried forward from one cell to the other and one generation of the cell to another. And during this process, because of the damaged repair process, the level of damage actually increases. And now, because there is a lot of DNA damage inside the cell, the chances of cell becoming cancerous will increase. So, these genetic defects have many consequences. So, some of them are listed below. So, some of the genetic defects which are associated with cancer can lead to abnormal signaling pathways. So, here because your DNA is damaged, now it is going to code for a protein which is again going to have a slightly different structure, and so this can lead to abnormal signaling pathways. It can also lead to insensitivity to growth inhibitory signals. So, as we discussed earlier, an intricate process of cell growth occurs where there are signals sent out to slow down growth, there are signals sent out to differentiate, there are signals sent out for apoptosis. So, when there is a signal given to inhibit growth, these genetic defects can actually make the cell insensitive. There is also another possibility that it leads to abnormality in cell cycle regulation. We will look at it later about what cell cycle is and how it can be abnormal. Genetic defects can also cause evasion of programmed cell death, which is also called as apoptosis. So, here when the cell is in such a damaged condition, it should normally trigger apoptosis, but now because of some of these defects that are present, it can evade this programmed cell death and become cancerous. And this essentially leads to limitless cell division, or what is known as immortality. But these cells cannot grow without the supply of nutrients. So, a major process in the growth of cancers is a process known as angiogenesis. So, here in angiogenesis, you need blood vessels to develop in the proximity of these tumors, so that you can have constant supply of nutrients. Lastly, these genetic defects can also cause what are known as tissue invasion and metastasis. We have already looked at previously that there are two types of cancers, one is a benign cancer and the other one is metastatic cancers. So, most if not all of these aforementioned conditions have to be met for cancers to develop. So, therefore, it is a fairly complicated disease where a number of things in a coordinated manner have to go wrong. However, it is quite common. So, for example, one of the mechanisms proposed is exposure to mutagen. Carbon in coal is suggested as a mutagen. There is also asbestos dust, which is quite harmful. So, it may take many years of exposure to the mutagen. So, for example, coal or asbestos it may take you several years for you to develop cancer. It is also possible there are a large number of smokers and tobacco consumers worldwide, and constant exposure to tobacco can accentuate this damage, right. So, once it accentuates it, what happens is that it overwhelms the safeguarding systems and develop into full blown cancer. So, the various hurdles and safeguards that a potential cancer cell has to overcome explains why cancers are relatively rare in early life, okay. And so, it becomes more common as we get older. It also explains why it is so difficult to treat once it appears. So, as there are so many cellular safeguards have already been overcome, it is unlikely that tackling one specific cellular defect is going to be effective. So, as a result, many anti-cancer drugs have tended to be highly toxic agents, okay. and they act against a variety of different cellular targets by different mechanisms. So, but being toxic, one of the problems is that they are quite poisonous to normal cells as well, and these end up causing a number of serious side effects. And these agents which are anti-cancer agents are said to be cytotoxic. Cyto means cell, toxic means bad or potentially killing, and the dose levels of these cytotoxic agents have to be chosen in such a way that they are high enough to affect the tumor, but quite safe for the patient. So, many of the side effects associated with these anti-cancer agents are because these agents are quite cytotoxic. So, cancer drugs are now being developed so that you can have them to be more selective, and these highly selective agents have potentially less serious side effects. So, given the number of defects in a cancer cell, a single agent might not work, and therefore, a combination of radiotherapy, surgery along with chemotherapy is what is typically necessary. So, let us look at some abnormal signaling pathways now. So, like I mentioned earlier, normal cells receive signals to grow, they receive signals to divide, and they receive signals to differentiate wherever the case might be. So, these signals that allow them to grow or propel them to grow are known as growth factors, and these are in many cases external cell chemical messengers which activate kinase receptors. So, you have a cell which with a let us say a kinase receptor, and so this ligand binds and it activates the growth. Okay. Now, these receptors will trigger a signal transduction pathway which eventually reach the nucleus to activate transcription of the protein, which are required for cell growth and division. Most, if not all cancers suffer from some defect or the other in this signaling process. So, therefore, the cell, instead of inhibition and growth after some time, it actually is instructed to multiply constantly. And as you can imagine, the signaling process is quite complex, and there are various points at which it can go wrong. So, many cancers are capable of growing and dividing in the absence of external growth factors. So, once cancer develops, it does not need any external growth factors, it just grows on its own. Right? They can also keep indefinitely growing by producing the growth factors themselves. Okay? So, therefore, you can have a series of cancer cells which are growing, which do not need any growth factor at this point, but then they can stimulate or they can produce these growth factors, which then further allows other cancer cells to grow. So, therefore, this is a process which can occur through autophosphorylation. So, through this mechanism, these cancers become immortal. So, the example here are platelet derived growth factors, which is called as PGDF, as well as transforming growth factor known as TGF alpha. Other cancers can produce abnormal receptors which are switched on constantly despite the lack of growth factors. So, for example, this ERB-B2 receptors in breast cancer cells. Right. So, these are always on, and therefore, you do not need an external ligand to activate it. It is also possible for the receptors to be overexpressed, and so you do not need a whole lot of the growth factor to be activating it. A small amount is sufficient for it to trigger growth. So, what it means is that the oncogene is too active and codes for excessive amounts of the protein receptor. So, once it is in the cell membrane, the cell becomes super sensitive to even extremely low levels of the circulating growth factor. So, there are many points where things could go wrong in the signal transduction pathways. So, for example, the RAS protein, which we have just looked at, is a very crucial feature in the signal transduction pathways, which lead to cell growth and division. Abnormal RAS protein is locked in the on position and is constantly active despite the lack of an initial signal from a growth factor. So, there are several external hormones which are used to counteract the effects of the stimulatory growth factor. So, these are what are known as growth inhibitory signals. So, one example here is the transforming growth factor beta, which is known as TGF beta, and these actually prevent the growth of cells and prevent them from dividing. However, cancers can become insensitive to these growth inhibitory signals. So, this is another way in which you can the cell becomes immortal. So, what happens is that when it becomes insensitive, then the risk of the cell becoming cancerous is quite high. So, this can arise from damage to the genes coding for the receptors for these inhibitory hormones, which are known as the tumor suppression genes. So, now let us look at some of the cell cycle regulation methods and how it occurs. So, during cell division, there are several phases which are involved. The first one is called the G1 phase, followed by an S phase. Subsequently, there is a G2 phase and an M phase. Now, these are highly coordinated events, okay, and they occur with great level of synergy. Now, during this process, a number of decisions have to be made by the cell. So, for example, when the G1 phase is initiated, so here is the G1 phase. Now, it has to decide at some point to go into S phase, right. So, there are chemical signals which are secreted to promote the growth, and it also helps, there are other signals which are produced to inhibit it. So, the G1 phase is where a cell is growing in size, okay, and it's starting to prepare to grow its to copy its DNA in response to various growth factors or internal signals. So, this is considered as the beginning of the cycle. Now, in the next phase, which is called as the synthesis phase or the S phase, is where DNA replication takes place. Once the cell's chromosomes are copied, then there is another interval which is called as the G2 phase or the gap 2 phase. In this phase, cell readies itself for cell division. Okay. So, this gap or interval is very crucial because it gives the cell time to check if the copied DNA is good, and if there is any repair DNA damage that occurs, then it is repaired. Okay. Now, finally, there is an M phase which is also called the mitosis phase, where the actual cell divides and produces two daughter cells, each containing a full set of chromosomes. So, the daughter cell can then enter the cell cycle again, or alternatively they may move into what is known as a resting state or the G0 state. So, there is a decision point here during this cell cycle which is called the restriction point, which is denoted by R. Okay. So, here this phase becomes abnormal in many cases of tumors. So, there are also various surveillance mechanisms or checking mechanisms which are known as checkpoints, which allow for checking of whether the genome integrity is still happening or not, is good or not. So, a delay will take place during the G2 phase, as we have discussed earlier, in case DNA damage is detected. And as we have discussed previously, cancers are frequently associated with damaged DNA, and therefore, one would argue that in the G2 phase there should be an arrest because there is going to be significant amount of DNA damage in the cell. So, now in this time that is given for repair, if the DNA damage repair is not possible, then there is another signal which is given for the cell to commit suicide. So, these checkpoints also become defective in tumor cells. So, for example, in this process there is a binding of a cyclin, okay, which is with its associated kinase, which activates the enzyme and serves to move the cell from one phase of the cycle to the other. So, for example, when a cell is in the G1 phase, a decision has to be made whether to move into the S phase and start copying DNA. So, this decision is taken depending on the balance of the various growth versus inhibitory signals that are being received through signal transduction. So, if the balance is towards cell growth, then there is an increase in cyclin D, okay, and this binds to the corresponding kinases CDK4 and CDK6. So, this happens in this phase, right. Now, so the resulting complexes phosphorylate a powerful growth inhibitory molecule known as PRB, okay, and this PRB, which normally binds and inactivates a transcription factor. Phosphorylation alters the PRB such that it can no longer bind to the transcription factor, and then the latter is free to bind to specific regions of DNA. Okay. So, this results in the transcription of specific genes which lead to the production of proteins capable of moving the cell towards the S phase. So, therefore, the cascade is initiated by cyclins which go and bind to CDK, which leads to phosphorylation, which then leads to transcription of specific genes which lead to the production of proteins that are capable of moving the cell towards S phase. So, the examples here are cyclin E and thymidine kinase. So, once cyclin E has been produced, it combines with CDK2 and that is shown here, right. And what happens is that this complex is now responsible for the progression of the cell from the G1 phase to the S phase. So, once other activated cyclin which is CDK complexes are important at different phases of the cell cycle. So, for example, the CDK 2 cyclin A complex, which is shown here, is very important in the S phase, whereas a cyclin B CDK1 complex is responsible in the M phase or the mitosis phase. Okay. So, restraining proteins are present which can modify the effects of cyclins. So, these include examples such as P15 and P16. Okay. So, what these do is that they block the activity of cyclin D CDK complex. So, another example of an inhibitory protein is P21, which is again controlled by P53, which we have looked at previously. So, P53 is an important protein that monitors the health of the cell and the integrity of DNA. So, here is P53 in this process. So, to summarize, progression through the cell cycle is regulated by sequential activation of cyclins and CDKs, and this process can be down regulated by CDK inhibitors, and the whole process is tightly regulated such that there is an accumulation of the relevant cyclin CDK complex followed by rapid degradation of the complex once the task is complete. So, overactive cyclins or CDKs have been associated with several cancers. So, for example, breast cancers often produce excess of cyclins D and E, and skin melanoma, which is a case where one develops a cancer on the skin, has lost the gene that codes for the inhibitory protein P16. So, keep in mind, you can have excess of the growth factors or you can have suppression of inhibitory proteins. Half of all human tumors lack a properly functioning P53 protein. So, P53 protein is the one that monitors the health of the cell, which means that the level of the inhibitory protein P21 also falls down. So, in viral related cervical cancers, both PRB and P53 proteins are often disabled. Now, with this, let us look at the induction of apoptosis. So, like I mentioned earlier, there is a self destruction process or suicide process that is programmed into each cell. So, once there is enough DNA damage that occurs and there is a huge problem with the genome integrity, usually apoptosis is triggered. This is the normal way in which the body protects itself. So, and this again is coordinated by a series of chemical signals. So, should there be any of these chemical signals that are absent, a self destruct mechanism is automatically initiated, as shown here. Okay. So, apoptosis is also important in destroying cells that escape from their normal tissue environment. So, for example, if in the case of of a muscle cell, let us say one of the cells is going to detach itself and go into the lung, the function of a muscle cell inside the lung is not probably useful. Therefore, what happens is that it is detected by our immune system and it is destroyed through the induction of apoptosis. So, cancer cells which metastasize have undergone a number of genetic changes that allows them to avoid this process. Okay. So, this could be a sustained lack of growth factors or hormones. So, you have these external influences that can clearly influence the growth, okay, and so by having this lack of growth factors, one could induce apoptosis. Or there are proteins which are known as death activator proteins, which can bind to the cell membrane, called as tumor necrosis factor receptors or TNFR. So, once these bind, then what happens is that it triggers apoptosis. The third way is, as I mentioned earlier, is through the immune system. So, once your muscle cell which has escaped goes into the lung, the immune system recognizes this as something that is not supposed to be there, and then the T lymphocytes perforate the cell membrane of the damaged cell and injects an enzyme called as the granzyme, which initiates apoptosis. So, there are also intrinsic routes that can result in apoptosis. So, one is the intrinsic pathway is due to DNA damage. As we have already discussed, the genome integrity and is really important, and once the DNA is damaged because of chemicals or drugs or oxidative stress, then what happens is that you need to trigger apoptosis. So, the cell monitors these systems, the integrity of the genome, and once it detects the damage, it leads to an increased production of tumor suppressor protein p53. At sufficient levels, p53 will actually trigger apoptosis. So, the process in which this happens is that p53, the various signals that are induced by p53 actually converge in the mitochondria, and the mitochondria contains what is known as proteins known as cytochrome C, and these cytochrome C actually secreted by the mitochondria, and the release of this from mitochondria results in a series of events which leads to the formation of what is known as an apoptosome. Okay. So, the process is, once p53 goes up, it results in interaction with mitochondria. Mitochondria then releases this cytochrome C, which then triggers the formation of a protein complex known as apoptosome. The apoptosome then recruits and activates an enzyme known as procaspase. Okay. So, procaspase 9 is a really important enzyme in the whole cascade of apoptosis. This in turn activates what are known as caspases. Okay. So, caspases belong to a protease class of enzymes, and these are cysteine proteases, and they contain cysteine residue in the active site, and they are important to the catalytic mechanism. We have already looked at cysteine based proteases previously. So, because these are proteases, what they do is that they destroy the cell's proteins, which leads to the destruction of the cell. Since caspases are so crucial to the cell, they are highly regulated. Okay. So, there are proteins which promote it, while there are proteins which also suppress it. The example of a suppressor is this BCL2 and BCLX. So, the relative levels of these proteins is dependent on the various monitoring procedures within the cell. So, once there is a high level of damage that is induced, then the levels of inhibitors of apoptosis may go down and the promoters of apoptosis may go up. Okay. So, now it is quite obvious for us to understand that these defects in the apoptosis mechanisms will have serious consequences. So, for example, in the case of radiotherapy and many chemotherapy regimens, both these processes occur by triggering apoptosis. However, if the apoptosis process is not well regulated, these molecules are not going to be, or these strategies are not going to work. So, this in itself will not be fatal to the cell. Okay. So, if the mechanisms involved in inducing apoptosis are defective, then the drugs do not become effective, okay, and the cell continues to be immortal. Okay. Now, with that, let us look at another important concept known as telomeres. So, when a cell divides and it forms two daughter cells, it can subsequently divide and so on, right. What happens is that this division process can occur a few times, maybe around 50 times or so, before they initiate apoptosis and die. But as I discussed, cancer cells evade this and they do not die, right. So, therefore, this division process, or the process by which certain number of divisions occur and after which they are going to stop, is mediated by enzymes, by structures known as telomeres. These are very crucial for the immortalization process. So, here is the telomere which is attached to the DNA. It's a part of the genome. Okay. So, chromatin we have already discussed previously consists of a protein which wraps around a variety of proteins, and telomere is a polynucleotide that is at the 3' end of the chromosome. So, these contain several thousand repeats of a 6 base pair sequence. Okay. So, the purpose of the telomere is to act as a splice for the end of the chromosome and stabilize and protect the DNA. So, after replication process, about 50 to 100 base pairs are actually lost from the telomere in this process. So, because DNA polymerase is unable to completely replicate at the 3' end of the chromosomal DNA. So, each time the cell divides, you lose about 50 to 100 base pairs. Eventually, the telomere becomes too short to be effective and the DNA becomes unstable, either unraveling or linking up with another DNA end to end. So, both of these processes are going to trigger apoptosis and the cell will die. So, during early stages of cancer, the presence of telomere is going to imply that the cell will divide only a few times, okay. But somehow the cancer breaks free from this process and becomes immortal. So, the cells maintain the length of the telomere by expressing an enzyme known as telomerase. So, this is a member of the group of enzymes known as RNA dependent DNA polymerases. So, telomerase has the ability to add hexanucleotide repeats on the end of the telomeric DNA, thus maintaining its length. So, this is an important process during development of an embryo, where telomerase is responsible for creating telomeres in the first place, but after birth the gene encoding the enzyme is suppressed. So, during the development stage in the embryo, the telomerase is actually active. However, after birth the enzyme is suppressed. So, immortal cells have found a way of removing that suppression such that the enzyme is expressed again. So, now telomerase enzyme is expressed in over 85% of the cancers. So, a number of efforts have been made to design drugs which will inhibit the telomerase, but so far there has been no success in developing it towards the clinic. Next we are going to look at another important concept, which is angiogenesis. So, as I mentioned earlier, the problem or the situation with cancers is that they need to have a steady supply of nutrients, including amino acids, nucleic acid bases, carbohydrates for constant growth and division, and of course, they need oxygen and growth factors. So, for all of this to happen, the tumor would need to have a good blood supply. So, as the tumor grows in size, its cell become increasingly remote from the blood supply. So, let us say you have a blood capillary network like this, and the tumor is located here. As it grows bigger and bigger, what happens is that the distance from this blood vessel goes higher and higher. So, this results in the center of the tumor having low oxygen tension. So, this situation is called as hypoxia. So, the cells in the center have less oxygen, and this triggers the buildup of an important protein known as hypoxia inducible factor, or HIF. An example of this is HIF1. So, these factors actually upregulate a number of genes that promote survival in oxygen starved environments. Normally, one would expect that if a cell is present in oxygen starved environment for too long, it would die. But this set of proteins known as HIF, hypoxia inducible factors, play a crucial role in the survival of cancers in these oxygen starved regions. So, for example, the growth factors are released from the cell, such as VEGF, which is vascular endothelial growth factor, and fibroblast growth factor, which is FGF2. These interact with receptors on the endothelial cells of the nearby blood vessels and stimulate these cells to divide, leading to branching and extension of the existing capillaries. So, what we were looking at earlier was the tumor is quite far away from the blood vessel. So, it secretes these factors which can go and stimulate the process known as angiogenesis, where branching and extension of capillaries can occur. So, these vascular growth factors are present in normal cells and are usually released when the tissues have been damaged. So, the resulting angiogenesis helps in repair of the injured tissue and is normally controlled by angiogenesis inhibitors such as angiostatin and thrombospondin. So, during injury these become important in order for the body to repair itself, and after that the suppressors such as angiostatin and thrombospondin suppress the angiogenesis and everything becomes normal. However, this balance is quite awry in the case of tumors. As a result, the tumors are able to receive the increased blood supply required for their survival. So, the chances of cancer cells escaping from the primary source and metastasizing are also increased, not only because of the increased availability of blood vessels, but also because of newly developing endothelial cells that can release proteins such as interleukin 6 that stimulate metastasis. So, the blood vessel arising from angiogenesis are abnormal, okay, and they are quite sort of disorganized in nature and they are quite leaky. Okay. So, these cells also display molecules known as integrins. Okay. These integrins appear on the surface of the cells and they are absent from mature vessels which protect the new cells from apoptosis. So, before angiogenesis can begin, the basement membrane around the blood supply vessel has to be broken. This is done by enzymes known as matrix metalloproteases or MMPs. So, inhibiting angiogenesis is a tactic which can be used to tackle cancers, and there have been drugs that have been developed to inhibit angiogenesis that break down these abnormal blood vessels. So, angiogenesis inhibitors in general are much safer and less toxic than traditional cytotoxic agents which are used in the clinic. Okay. But these cannot work on their own because what they will do is only to suppress certain kinds of certain parts of the tumor will not grow, but you can give it along with traditional cytotoxic agents to completely kill the tumor. Okay. So, this normalization can help anti-cancer agents reach tumors more effectively. In the longer term, it serves to stall tumor growth, then shrink it by breaking down abnormal capillaries. So, as a result, the tumor becomes starved of nutrients and growth should decrease. So, these are some of the advantages of inhibiting angiogenesis. So, some anti-cancer treatments take advantage of the leaky blood vessels which result from angiogenesis. So, since the tumor is quite disorganized, there are number of leaky blood vessels in this. So, what we can do is we can encapsulate these cancer drugs into what are known as liposomes or nanomaterials or drug delivery systems, which are too big to escape from normal blood vessels, okay, but they can actually escape from these leaky vasculature that is present in tumors. So, the anti-cancer drug is actually concentrated at the tumor. As the tumors do not generally develop an effective lymphatic system wherein this is kicked out, these polymer drug delivery systems tend to be trapped at the tumor site. So, even with angiogenesis, there are regions of well developed tumor which fail to receive an adequate blood supply, and as we discussed earlier, these are known as hypoxic regions. Okay. So, as most anti-cancer agents act best on actively dividing cells, this becomes a huge problem. So, anti-cancer therapy will be successful in halting a cancer and eliminating most of it, but once the treatment is stopped, these cells which are in the center or they are dormant, they can start multiplying and the tumor reappears. The problem with these kinds of cells is that they are also more likely to metastasize. So, hypoxic tumors are, in order to survive, will have to revert to anaerobic respiration, which is glycolysis, and this results in a large buildup of acidic byproducts. The cell addresses this problem by exporting acidic protons into the extracellular space. So, once there is a lot of acid that is present, so it exports the H plus out. Okay. So, because of this, the tumor environment tends to be more acidic than normal tissues. So, a number of anti-cancer therapies have attempted to take advantage of this difference in pH. So, for example, one could think about selective localization of porphyrins, which we have discussed previously in photodynamic therapy. The next concept that we will look at is tissue invasion and metastasis. So, like we discussed earlier, there is a large buildup of hypoxia inducible factor in oxygen starved environments. So, of course, not all cancers are life threatening. So, there are a number of benign tumors which are highly localized and they can grow to the size of a football without any fatality. But malignant cancers, on the other hand, are life threatening, because these cells have the ability to break away from the primary tumor, invade a blood vessel or a lymphatic vessel, travel through the body and set up tumors elsewhere in the body. So, in this process, there is a number of changes or number of cellular processes that need to occur. So, cells have a molecular signature on their surface which identifies whether they are in the correct part of the body or not. So, like we discussed earlier, a muscle cell is if it escapes and gets to another part of the body, then the immune system is able to recognize it. So, there are molecules known as cell adhesion molecules or E-cadherin, which ensure this to occur. So, these help in cells adhering to the cells of similar character, okay, and they form an insoluble meshwork of protein filling the space between them, known as the extracellular matrix. So, this is true for epithelial cells, that is the ones lining the gut, lung, etc. So, adhesion of the extracellular matrix is particularly important because it is necessary if the cell has to survive. So, here there are molecules known as integrins which are involved in the anchoring process. So, you have here is the cells which are anchored to a surface, and then there is a intricate mesh of E-cadherin that is formed, and then there are molecules known as integrins which are involved in the anchoring process. So, if a normal cell becomes detached, that is, let us say this is it becomes detached. So, here is your other cells. So, this is the detached cell, right. Now, because it presents very different, if the normal cell becomes detached, it stops growing, okay, and after some time this apoptosis is triggered. So, this is because the normal cell can survive only if their addition molecules match the extracellular relevant molecules. So, here is your cell surface, your tissue surface, here are your cells, and so unless there is a matching of these two, it does not survive, right. So, therefore, if the matching process becomes incorrect, it triggers apoptosis and the cell dies. So, this is one of the ways in which our body ensures that there is no transfer of inappropriate cells across the body. So, in the case of tumors, these cell adhesion molecules are missing, and so this allows for them to break free from the tumor and metastasize. So, these cells are called as anchorage independent cells. So, here is one type of anchor and here is another type of anchor. So, the tumor can attach itself here, it can metastasize and attach itself here. So, therefore, these tumors become anchorage independent, and that this allows for this tumor to grow, and this is the process of metastasis wherein the tumor starts to spread. So, oncogenes which are present in these cells or which code for proteins which send what are known as false messages back to the nucleus, implying that the cell is detached. Okay. So, it is noticeable that most cancers derive from epithelial cells. So, once an epithelial cell has gained the ability to split away from its neighbors, it needs to gain access to the blood supply if it is to spread around the body. However, epithelial cells grow on a basement membrane, that is a thin layer of extracellular matrix which acts as a physical barrier to the movement of cells. So, there are only two types of cells which can breach this physical barrier. One is cancer cells and the other one is white blood cells. So, white blood cells are needed, white blood cells need to breach this barrier because they can reach the area of infection and address it. Whereas, when cancer cells breach this barrier, they spread the disease. So, both of these require the use of matrix metalloproteases, which we have discussed previously. So, these matrix metalloproteases which will help with hydrolysis of this barrier. So, once a cancer cell breaks through the basement barrier, it has to break down a similar barrier surrounding the blood vessel in order to enter the blood supply. So, for a cancer which it has to cross this epithelial layer, and then there is a blood vessel that it has to cross, breach, and then it has to metastasize. So, statistically less than 1 in 10,000 cells succeed in setting up a secondary tumor, but the problem is all we need is one cell to do this, and so then metastasis has occurred. So, these circulating tumors get trapped in a network of capillaries and then set up secondary tumors. So, here is your blood supply in which your tumor is going, and then what happens is that there is a network of capillaries, and here once it gets to a network of capillaries, it lodges itself there and then starts growing and starts making a secondary tumor. And for many of these, the lungs turn out to be the focus for secondary tumors. So, in the case of cells originating from the intestines, it is the liver. Some cancers produce factors that cause platelets to initiate blood clotting around them, such that they increase in size, become stickier and stick to the blood vessel, which allows them to escape.